Hi. Guys, if you're with me today, say hello in the chat. Let me know who we've got for our lesson, our second year 13 lesson of the week. Say hello. Plan for the lesson today is we're going to go back to GCSE and think about some genetic crosses and some key genetics terms. And then we're going to build on it and introduce the new year 13 content, which is our dihybrid crosses. So how to do quite a complex genetic cross. Who's here? Guys, why is no one in the chat? What is going on? I don't want to start without you. So let me know if you're here and let me know if you can hear me. Hi, Amberjeets. How are you doing? Are we ready for some genetics? Hey, can everyone hear me okay? I think the genetics unit in year 13 is one of my favourites, I've got to say. Oh, I'm good, thanks too. Yeah, I'm good. Yeah, as I said, I'm looking forward to this lesson because this is one of my favourite units. The reason I like it is because it's less content heavy, right? We all know biology is very content heavy. There's so much to learn in every module. But with this one, it kind of stands out as being a little bit different to the others because it's less about learning content and more about doing these little puzzles, these Punnett squares and genetic crosses. So once you've kind of got the knack of doing that, the questions are quite easy. Dyed your hair? I haven't actually, it's just a little bit wavy. So maybe that's why it looks different. Okay, so the little starter activity that I've got for you today, we're going to try and get our key genetics definitions in. Now, you have not done this at all in year 12. This is a GCSE topic and we don't really do it again until year 13. So if you can remember any of these definitions all the way back from year 11, put them into the chat. Okay, you can even just pick one or two. And then we're going to write some like perfect definitions because usually on the A-level exams, they'll ask you for at least one of these, yeah? One of your marking points will likely be a genetics definition. Oh, thanks, Saya. Okay, so let's go. Just pick one or two, whatever you feel like confident with, and put it into the chat for me. Perfect. Right, I'm going to take yours, Aaliyah. So Aaliyah's already done a perfect definition for gene. So I'll write that in. I suppose we did go over this one in year 12 as well as GCSE. I like the way you said polypeptide as well. I mean, you can get away with protein, but polypeptide is really good because it is the DNA that determines the order of amino acids, isn't it? In the polypeptide chain. Obviously, some proteins have multiple polypeptide chains. So polypeptide is a better word, really, if you can use it. Allele, we've got allele going on as well, different versions of a gene. That will do. Different versions or different forms of a gene. Anything like that would be sufficient. So we're going to look at allele first and just make sure that we're clear on what that means. Grace went for locus, the location of a gene on the double helix. Or what we normally say, Grace, is just the location of a gene on a chromosome. That will be what it says on the mark scheme, okay? The location of a gene on a chromosome. Let's see, has anyone got dominant? Good, Annie says dominant, the allele is always expressed. I'm going to elaborate on that a little bit and say the allele, if it's dominant, is always expressed. I'm just going to make it even better. The allele is always expressed if present in the genotype. Okay, so if you have a dominant allele in your genotype, that allele will always be expressed. Let's have a look. Priscilla, genotype, genetic makeup of an organism. Yep, good. I'll do recessive first, I think. I can't see anything for recessive. We left that one out. We're not sure what the best definition is. Let's go for homozygous then. because you've done that one, having two of the same alleles for a gene, or like you said in the chat, having two identical alleles for a gene. Good, Annie. Whereas heterozygous is having two different alleles for a gene. Have we remembered this pretty well, would you say, from GCSE? I'm just interested to know, is it one of the topics at GCSE that you have remembered? It seems like you've remembered them quite well. So let's go back to what Priscilla said for genotype. I'm going to say the alleles an organism has for a gene. We normally express a genotype as 2 letters, right? Like, I'll just give you some examples from GCSE. Like, you'll have seen genotypes represented like this. It's just the alleles that you have for that gene. It might be 1 dominant, 1 recessive, or 2 recessive, homozygous or heterozygous. Phenotype, the observable characteristic determined by both genes and environment. These are perfect definitions. How are we getting these so good? Because a lot of GCSE students would just say observable characteristics, but Jay is spot on. It's the observable characteristics determined by both your genes, or you could say your genotype, and your environment. Good. We did a little bit when we were studying mutations. Yeah, I suppose when you're doing mutations, you go back over like genes and alleles and things like that, don't you? So let's go back to recessive, because I noticed Tempered Steel and Felicity. Yeah. So Tempered Steel says recessive is a type of allele that needs 2 of those alleles to be expressed. Yes, that would be fine. Felicity's worded it slightly differently, but that's fine as well. It's only expressed if there's no dominant allele present. Yeah, both fine. I'm going to go for the allele is only expressed if there is no dominant allele present. Now, at A level, you're going to learn about some other examples. You're going to learn about codominant alleles. Does anyone have an idea what codominant alleles might mean? I might just add it on. I mean, the name kind of gives it away. What do you think codominant alleles means? Codominant. Any suggestions? Doesn't matter if you get it wrong. Yeah, good, Grace. Spot on. Yeah, 2 different alleles that are both dominant, both expressed. Yeah. So you didn't do that at GCSE, but you will be doing that at A level. So in flowers, for example, if they have both a red and a white allele, the flower ends up looking pink because the red and the white are codominant and they both get expressed and the flower looks pink rather than just being red or white. Yeah, perfect. Okay, have we had time to get these written down? Or do you want me to just pause on the screen for a second? Let me know in the chat. Are we good? Are we good to move on? Because we're going to start now doing some simple genetic crosses and we're going to build it up to an A-level year 13 genetic cross. We're good. Perfect. Okay. And remember, you can always go back and watch these videos again if you need to just go back and watch this or pause the screen. Okay. So this is really simple. I'm sure you guys will all be happy with this, but just to go over the basic idea of an allele. All individuals of the same species have the same genes, but not necessarily the same alleles. So think of us as humans. We are all humans, yeah? Hopefully. We all have the same number of chromosomes. We all have the same genes. But we have different versions of those genes. That's why we all look different and have different personalities, okay? So it's not that we have different genes, it's just we have different versions of them. Now I'm using this diagram just to go over a really basic example, which is eye colour, yeah? And it gets us to practice our key terminology. So this individual A, is heterozygous for the eye colour gene. So you can see they've got one allele for blue eyes and one allele for brown eyes. Heterozygous. Homozygous, these alleles are both brown, and homozygous recessive, so these alleles are both blue. We all know that brown is dominant, right? So this individual that's heterozygous, because they've got a dominant brown allele, they will have brown eyes because the dominant allele is always expressed if it's present, yeah? So that first individual there would have brown eyes, individual B would have brown eyes, individual C has got blue eyes because there's no dominant allele. So the recessive allele is expressed. Easy peasy, no bother. So what I want you to do, this text has come up a little bit funny. Just tell me, have a look at the genotype. So this is genotype 1, this is genotype 2, and this is genotype 3. Obviously, these are meant to be next to each other, guys. I'm sure you realise that. I think that it's just shifted the letter down, hasn't it? Just give me the phenotype super quickly in the chat. So give me the phenotype, the observable characteristic that is expressed. Give me the phenotype for person 1, 2, and 3. Just do that super quick. I'm just checking your basic knowledge here. Good. So Daisy says brown, brown, blue. Exactly. Okay, so the phenotype is always what we actually see. It's the observable trait. Whereas the genotype, as we said before, is the alleles we have for a gene. Okay, so this individual here is heterozygous. How would we describe this genotype? If they have 2 dominant alleles, how would we describe this genotype? What do we think? 2 dominant alleles. How would you describe the genotype? Homozygous. Can you add anything else on, Aaliyah? Homozygous what? Good, Zaina. Good, Daisy. Homozygous dominant. Just to differentiate, exactly, because you could be homozygous recessive, like this individual here is homozygous recessive, because they've got 2 recessive alleles. Again, easy peasy. So let's do a genetic cross. You're going to think that I'm really old-fashioned here because I've not done it as a Punnett square. I've done it in the way that I was always taught to do it. But we can do a Punnett square as well if you prefer. And I'm sure you've heard of Gregor Mendel. Who studied Mendel at GCSE? What do we know about Mendel? He was the father of modern genetics. Do we know anything else about Mendel from GCSE? Gregor Mendel. I did, and I did not like him. Daisy says pea plants. He loved the pea plants. He was wrong. Why was he wrong? Yeah, he was obsessed with peas and doing breeding experiments with pea plants. Oh, the Mendelian ratio. Yeah, Henry. That's the ratio for a dihybrid cross, which we're going to do today, actually. For a simple monohybrid cross, if both parents are heterozygous, we get a 3-to-1 ratio. Yeah, he was a little bit weird, maybe. But yeah, he was a monk and he spent a lot of time in the monastery gardens just breeding peas, looking at the outcomes. And he basically figured out that traits were being passed on. And then he figured out the idea that some traits were more dominant than other traits. Yeah, your school taught you a lot about him. Yeah, I mean, it was on the spec for GCSE. So we're going to try and breed a plant that was pure tall. Now, what I mean by pure tall is homozygous for the tall alleles, yeah? And pure short, again, homozygous, but for the short alleles. What letter could you choose to use to do this genetic cross? What letter would you choose? If you were left to choose your own letter to represent these alleles, what letter would you choose? So you're going to choose two different letters, are you? Aaliyah just says T. I wouldn't choose X and Y because X and Y we always use for the sex chromosomes that determine sex. I would just use one letter because these alleles are for the same gene. Yeah, I would probably use T and T. Do you remember this? You just use one letter because these alleles are for the same gene. They're for the height gene. You could use H for height, but you're just going to use one letter and you're going to differentiate by using the capital letter for the dominant and the lowercase letter for the recessive. But we must use the same letter because these alleles are for the same gene, the height gene, okay? You can choose any letter, by the way. If they don't tell you what to use, you can choose any letter. But my advice would be, don't choose a letter that looks the same when it's capital versus lowercase. So T is quite a good one. H is quite a good one. I wouldn't use S because S kind of looks the same, right? Whether it's capital or lowercase. So the genotypes of the parents. These parents were homozygous. So homozygous tall and homozygous short, okay? I haven't actually told you in the question, but I know that tall is dominant. So I've used the capital letter for tall. Then the genotypes of the gametes. Now remember, gametes, this is kind of where I want you to understand this now. You might not have understood this at GCSE, but gametes are made by meiosis. So they only contain single chromosomes. They don't contain pairs. They contain single chromosomes. Remember, a gamete is haploid. It contains single chromosomes. So you wouldn't have two alleles for the same gene in a gamete. You'd only have one. Yeah, because we separate those pairs of chromosomes. So the gametes will only contain one copy of that gene, one allele for that gene. Okay, this parent will obviously make gametes with recessive alleles in. Then we do the genetic cross. Now, you guys have probably learnt it like this. Let me know in the chat. Did you all just do Punnett squares at GCSE? Did you do it like this? Yeah, you all did Punnett squares. That's absolutely fine. We can do Punnett squares as well. You can do Punnett squares at A level. It's absolutely fine. But you can see what I'm doing. I'm using arrows. I just say like, well, this could pair with this, or this could pair with this, or this could pair with this, or this could pair with this. Okay, I'm just showing you which gametes could fuse together at fertilization, because one of this parent's gametes is going to fuse with one of this parent's gametes at fertilization. So this is how I was taught to do it, because I kind of used to go, well, this gamete could fuse with this gamete, yeah, or this gamete could fuse with this gamete. That's how we used to do it. So phenotypes, guys, in the chat, tell me, what is the phenotype of all of these offspring? Because the genotypes, they are all heterozygous. So what would the phenotypes be? Yeah, easy. They're all tall, which is obvious, right? Look, if you've got a parent plant that is homozygous for that dominant allele, all the offspring are going to get the dominant allele, so they're all going to be tall. Easy. And we'll do another Punnett square for the next one rather than doing it this way if you prefer. So now we're going to cross the offspring. Okay, so just go back for 1 second. We're going to cross two of these offspring. Okay, so this would be what we call making the F2 generation. So here, the offspring are referred to as the F1 generation. So this is the first generation of offspring, the F1 generation. Now we're going to cross two of those offspring plants. We're going to create the F2 generation. Who's heard of that before? F. Can't actually remember. Is that bad? I did know, but I've forgotten. I don't know if Neve's in the chat. Does Neve know? Anyway, so we're taking two of the offspring. So the offspring were tall, but the offspring were both heterozygous this time. So the gametes, each of those parents can make two different types of gamete. Some of the gametes will contain the dominant allele, and half of the gametes will contain the recessive allele. Then I do my little weird way. But you can do it like this. So you put one parent's gametes at the top, you put the other parent's gametes at the side, and then you show all the combinations that will result at fertilization. Okay. Filial. I was going to say something like that. Filial or finial. I couldn't remember. What does it mean, Annie? So give me the phenotypes then, please. By the way, it doesn't really matter which way you write these. You might be thinking, why have I written it that way round? It doesn't matter. I could have written it that way round. Good. Tall, 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 short, or a quarter are short and 3/4 are tall. Yeah. So we just write the phenotypes out like this. And then we might be asked for a ratio. So it's a 3-to-1 ratio, isn't it? Of tall to short, or we might be asked for a percentage. It depends on the question. Yeah. Filial means daughter. Oh, that makes sense then, doesn't it? That makes sense. So F2 generation here. This is our F2 gen. Okay, so yeah, they'll ask you for either a ratio usually or a probability. If they just say probability, you can even give it as a fraction like this or a percentage. Okay, still finding this easy. I hope we are. It is going to get harder. I'm just easing you in as usual. It is going to get harder, but I'm trying to make sure you've got the basics really solid so that when it gets harder, you find it easy. Because I think this is easy. If you get it, it's easy. Okay, we're going to do another one, just quickly. Okay, we're going to do F1 and F2 generation on the same slide. So we're going to cross, we've got purple and white. Now I'm going to tell you that purple is dominant and white is recessive. I can actually see that in the diagram because when we cross a purple and a white, it tells us that all the plants produced had purple flowers. So purple must be dominant, right? So I didn't really need to tell you that. What letter are we going to use here, guys? I'm going to avoid P and W because P and W would look the same, capital, lowercase. So I'm going to avoid that. How do they know a parent is heterozygous and not dominant homozygous? Well, if you have a look at this one, for example, Saya, all the plants produced had purple flowers. Now that tells me that this parent plant must have been homozygous dominant. Yeah, because if that parent plant was heterozygous, then we could have produced some white flowers in that first generation. Does that make sense? Should we go for D? Doesn't really matter. F for flower. Fs can look a little bit similar though when I write them. So I might go for D just randomly. Let's go for D. So I'm going to use D for purple and I'm going to use lowercase d for white. I'm using the same letter because they're the same gene, both the gene for colour, just different versions of it. So I'm going to cross my homozygous dominant purple flowering plant with my white. Now white is recessive, so I know this plant must be homozygous recessive because to be white, they would have to have 2 copies of that recessive allele. Okay, then I'm going to show the gametes that can be produced and I'm going to put them in circles just to show that they're gametes, which is something that we quite often do It's quite good practice to do that so that your diagram is clear. Then I'm going to do my Punnett square to show the combinations for the F1 generation. And I'm going to see here that they're all going to be purple, which is what the diagram told me. Okay, and they're all purple because the purple parent was homozygous dominant. So all the offspring got a purple allele, yeah? But now I'm going to cross 2 of the offspring and I'm going to get an F2 generation. So I'm going to take 2 of these heterozygous offspring. I'm going to show the gametes that they will produce in meiosis. And remember, gametes are haploid, so they'll only have 1 of each chromosome and therefore only 1 allele for each gene. I'm going to do my Punnett square. This is still just really simple stuff, yeah? I'm going to work out the combinations at fertilization. And all I want you to do is give me the ratio, please. So Grace has given me the probability. What is the ratio? Good. 3-to-1 ratio, which is the standard ratio, okay? I'm going to just tell you something now that maybe you didn't realize at GCSE. If both parents are heterozygous, if both of them are heterozygous, we always get a 3-to-1 ratio when we do a genetic cross. You might not in reality, when you actually do the breeding, you might not get a perfect 3-to-1 ratio in your offspring, but when you do a genetic cross, like on paper, when both parents are heterozygous, you'd always get a 3-to-1 ratio. So sometimes you don't even need to do the cross. If both parents are heterozygous, you just know it's going to be 3-to-1. Can I ask you a question? In reality, if we breeded these plants, why might we not get a perfect 3-to-1 ratio in our offspring? If we actually breeded these plants and we looked at the offspring, why might it not be a 3-to-1 ratio, purple to white? Daisy says environmental factors. Possibly, but let's imagine that this trait is purely controlled by genetics. We still might not get a 3-to-1 ratio. Good, Aaliyah. Aaliyah says the gametes that fuse are random. Genetics is random. Chance, yes. The little phrase that we use, which you should try and remember, is fertilization is random. Okay. It's just like with males and females, right? Every time a couple has a baby, there's a 50% chance of it being male and a 50% chance of it being female. But some couples might have 6 sons or 8 daughters. It's random. Same with tossing a coin. Yeah, you expect if you toss a coin 50 times, you'd predict 25 heads and 25 tails, a 1-to-1 ratio. But you might get 40 heads and 10 tails because it's random. It is down to chance, okay? So these are just predicted ratios, but in reality, fertilization is random, okay? And Saya is kind of thinking of something else as well. In meiosis, there can be crossing over and there's other things to think about. But for now, we're just going to say fertilization is random because that is usually a 1-mark question, okay? Okay, we're going to do some dihybrid crosses. Has it got anything to do with crossing over? Not in that example, no. But this is really confusing now. Not in that example, but sometimes if the 2 alleles are on the same chromosome and crossing over occurs, the allele combinations can get mixed and then that would affect the ratio. But it's only relevant if both of the alleles are together on the same chromosome, which is another topic. That's one of the hardest topics that we do in year 13. Okay. Right. So Mendel extended his work by looking at the inheritance of two characteristics at the same time. This is called dihybrid inheritance. You did not do this at GCSE. At GCSE, you only looked at monohybrid inheritance, which is where you just look at the inheritance of one gene at a time. Yeah, like you just look at the colour gene or the height gene or the eye colour gene. You just look at one gene at a time. But what you can do is look at the inheritance of two genes simultaneously. And this would be called dihybrid inheritance, okay? And you have to do a much bigger Punnett square, okay? So much bigger. You can see this Punnett square here is a 4x4 Punnett square. Now, we're not really going to do anything on this slide. This is just an example. We're going to do some on the next slides. Can you do it with 3? You probably could, but we never go beyond 2, okay? You could probably try doing it with 3, but we only ever do it with 2. At A level anyway. I'm not sure about university. So in this example, Mendel was looking at the inheritance of two genes. The gene for pea colour and the gene for pea texture. We know he was obsessed with peas. Yeah, who knows why. But he's looking at those genes together. So he looked at the colour. Now, yellow was the dominant colour, green was the recessive colour. I hate that we've used the letter Y here because the Y looks exactly the same, whether it's capital or lowercase. But he also looked at the texture or the shape. They've described it as texture/shape. So rounded peas, they were dominant, but the wrinkled texture, the wrinkled peas, that was recessive. And we're looking at them both together. So have a look at this diagram. What they've done here is they've written the genotype for each pea plant, but they've shown the alleles for both genes. So this dude here, just do it a bit bigger down here. This yellow pea was homozygous dominant for colour and homozygous dominant for seed shape. So this pea plant would look yellow and round. Okay, but it was crossed with a plant with this genotype. So this plant here is homozygous recessive for colour, so it would look green, and homozygous recessive for texture, so it would look wrinkled. Okay, do you understand this so far? We're going to do an example now, I think. It's basically exactly what we've already done. It's just that you have to work out the full genotype. So you're looking at two genes, you put them next to each other, and you're going to do a much bigger Punnett square. Okay, I think we should just get into it because the way to learn it is to do them really. So let's just get into it. So we are going to cross a tall pea plant. Let me just, so tall is dominant, obviously. So a tall green pea plant is crossed with a tall green pea plant. Okay. So we've got, do they both have the exact same genotype? No, they don't. Okay, so it's going to be quite interesting. They've not got the same genotype, even though they are both tall and green, they do have different genotypes because this one is homozygous for the tall allele and this one is heterozygous for the tall allele. Okay, but they're both tall because tall is dominant. And then this one is heterozygous for the colour allele and this one is also heterozygous for the colour allele. Now, I think the most complicated bit is figuring out the gametes. Now, I can give you a really quick way to do it, but I do want you to understand it as well. So I'm going to show you the really quick way, and then I'm going to try to explain why I've done it. Now you need to follow my pen on the screen to figure out the gametes, okay? Is everybody watching? So what I'm going to do to figure out the gametes is I'm going to say this allele could pair with this allele. And then I'd have this gamete. Or this allele could pair with this allele. And then I'd have this gamete. Or this allele could pair with this allele, and then I'd have this gamete. Or this allele could pair with this allele, and then I'd have this gamete. And basically just figuring out all the possible combinations, okay? Because remember, and this is where I want you to understand it, during meiosis, chromosomes are separated, okay? So the T's are going to get separated and the G's are going to get separated. Now they will then be put into the gametes in any combination, but you can only ever have one allele for each gene. So the dominant T could end up with the dominant G, or the dominant T could end up with the recessive G. Or this dominant T could end up with the dominant G, or this dominant T could end up with the recessive G. Okay? Question, why aren't the gametes pairing with the... Because we're just not onto that step yet, Saya. We're going to do that in a second. Okay? All I'm doing so far is figuring out which gametes can this parent produce in meiosis. So we'll do the same for this parent. So again, follow my pen. This allele could pair with this allele. So this parent could make those gametes. Or this allele could go with this allele. And we'd have some of these gametes. This allele could go with this allele. And then I'd have those gametes. Or this allele could go with this allele. And then I'll have these gametes. Okay, so each parent can make 4 different combinations, 4 different gametes. Okay, how do we feel about that? Because all we do next is a Punnett square, where we put one parent's gametes at the top, the other parent's gametes at the side, and we look at all the possible combinations. Okay, so let's go for it. Now, I could do this. There are 4 gametes that this first parent can produce. Is there any point doing this? Saya says, why is each parent combining with itself? I'm not crossing a parent with itself. I'm just showing what gametes that parent could make. So when, these are plants, right? So when this first parent makes pollen during meiosis, I'm figuring out what alleles could be in the gametes. So I'm not doing the cross yet, Saya. I'm just figuring out the gametes produced by that plant. Okay, is there any point in doing this? Param says no. There's no point if you do it without the copies, you guess. Yeah, there's no point because look, these 2 gametes are the same and these 2 gametes are the same. So I'm just going to be repeating myself. So there's no point because I can see that 2 of the gametes are the same and 2 of the gametes are the same. So there's no point. I may as well just do this. Makes it much simpler for myself, okay? What about this guy? Are all these gametes different? They are, aren't they? All those gametes are different, I think. So I am going to have to write all 4 of those ones out. There are 4 different gametes. Yeah, exactly, Saya, because we're looking at 2 different genes and each of those genes is only going to be 1 allele for that gene in the gamete. And we don't know like which allele for which gene is going to go into which gamete. So we're working out all the possible combinations that could be in the gametes. Okay, so once I've done this, I've got one parent's gametes on the top of my Punnett square. And because that parent only actually made two different gametes, I've only had to write 2 on the top, but I've had to write all 4 for the second parent because the second parent made 4 totally different gametes, okay? Now, when I do my Punnett square, I am going to put the letters back together in their pairs. So I'll show you what I mean. Can you see I've fused this gamete with this gamete, but I have put the alleles back together so they're in their genes. Okay, try and do the rest before I do it, guys. Try and do it in your workbook. You've got to be so careful not to make mistakes. It's so easy to just make a mistake when you're doing this quickly, especially in an exam, I'm sure. Okay. Now, the tricky thing is we need to work out the phenotypes, which I'm going to try and squeeze in the boxes, I think, rather than writing it all out at the bottom. So tall was dominant. That means short is recessive. I'm just going to write this at the top so we know what the recessives would look like. Green is dominant, so I'm going to say yellow is recessive. Okay. So they are all tall. Should we put that in first? Tall, tall, tall. And you know what? Just looking at the parents, you should have been able to tell that they would all be tall because parent 1 was homozygous for the tall gene. So obviously all the offspring are going to get a dominant tall, right? So they're all going to be tall. Are they all going to be green? Because we have to write both parts of the phenotype in. Grace says no. Yeah, we've got a yellow here. Ooh, and a yellow there. Okay, Grace says 2 are yellow. Yes, I got the same as you, Grace. What's the ratio then, guys? What's the ratio? What's the phenotypic ratio or the ratio of phenotypes? Grace says 6 to 2. Yeah, you simplified it there before I even asked. Thank you. So you've actually got a 3 to 1 ratio. So we've got 3 to 1 ratio of tall and green versus tall and yellow. Okay. Should we try another one? How do we feel about this? Can you give me a score? Give me a score from 1 to 10. 1 is, I haven't got a clue what you have just done. And 10 is like, I love it. I'm really confident. We've got some high scores. High scores. This is good. This is really positive because some students never get this. I'm trying to take it slow with you so that like you really understand what we're doing. I hope I'm not sounding patronizing. What's the purpose of doing this though? You'd ask that about anything. In terms of your biology A level, I think it's testing your logic, right? It's a bit of logic, but it's also testing you understand the process of inheritance, making gametes, fertilization. But in terms of Mendel and his peas, who knows? So what does it show? It shows that if you cross these two parents, we would expect to get a 3 to 1 ratio of tall green versus tall short. That's all it shows, really. Right, this is a bit of a harder one because I've not given you the parental genotypes. You have to work them out from the question. So we're looking at 2 genes. So we're looking at spotted skin. That's one of the genes. And we're also looking at woolly hair. What a random combination. This is the kind of combinations you get in the exam. So Oh, so it's like a normal Punnett square, just with the full parent's genes. It's like a normal Punnett square that you did at GCSE, but you're looking at the inheritance of 2 genes simultaneously, just to try and step it up and make it a little bit harder for you. In the main course, would you go over this again? Yeah, so in the main course, like, we tend to have a lesson on each topic, so there'll definitely be a lesson on dihybrid inheritance, yeah. But we'll do far more examples. Like today, we've only got an hour, so I think I've got 3 questions. In the main lesson, we'll just do loads, and we'll do some of the harder ones as well. So assume that spotted skin. So spotted skin is dominant. I like making notes as I'm reading the question. And woolly hair is dominant over non-woolly hair. This is a random question. Right, cross a heterozygous spotted non-woolly man. Can you put that genotype into the chat, please? Because this is harder because you have to figure out the genotype. Heterozygous spotted, and we're using S, so we're going to have to be super careful how we write it. Heterozygous spotted non-woolly man. Oh, we're using W. This is awful. I'm going to have to be really careful how I write this. Okay, so heterozygous spotted non-woolly. Non-woolly is recessive, so he must have 2 recessive alleles. And then we're crossing that person with a with a heterozygous woolly haired non-spotted woman. Non-spotted would be recessive, right? So that's what we're doing. I really want to change these letters. This is going to drive me mad. I'm going to have to make the size really obvious. Okay, let's work out the gametes. Yeah, good. Good, Saya. Let's work out the gametes. So remember, only 1 allele from each gene can go into the gamete. So we need 1 allele from the spotted skin gene and 1 allele from the woolly hair gene, but we need to work out all the possible combinations. So this allele could go with this allele, and we'd have this combination. This allele could go with this allele. That's exactly the same, but I'll write it, seeing as we're quite new to this. This allele could go with this allele, or this allele could go with this allele. They're exactly the same, but I'm going to write it just so you can see. Good. Do the same on the other side for me, guys. So this allele could go with this allele, or this allele could go with this allele. This allele could go with this allele. That's exactly the same. Or this allele could go with this allele. That's exactly the same. Okay. So because this parent was homozygous for the woolly gene and this parent was homozygous for the spotted gene, we don't have 4 different gametes. So we don't have to do as big a Punnett square. Okay, there's no point writing out repeated gametes. So I'm just going to write these gametes for parent 1. There's no point writing out all 4. And then I'm going to do, trying to make my letters look different, those gametes for parent 2. And then I'm going to do what would happen at fertilization. These letters are driving me mad. I should have used different colours or something. And remember, I put the alleles back together next to each other in their genes. Okay. Yeah, it's going to be a 2 by 2. There's no point doing a 4 by 4 because you would just end up repeating the same combinations. So what have we got? This one here, spotted skin is dominant. So this one is spotted and is woolly dominant. Yeah. So this one is spotted and woolly. This one is non-spotted and woolly. What are we even talking about here? This one is spotted and non-woolly. And this one is non-spotty and non-woolly. woolly. Do you think it's a 1 to 2 to 1? I didn't get that, I don't think. I've got spotted and woolly, non-spotted and woolly, spotted and non-woolly, non-spotty and non-woolly. I've got a 1 to 1 to 1 to 1 phenotypic ratio, or ratio of phenotypes. Yeah. Do you get that, Jay? You see it's a 1 to 1 to 1 to 1. They're all actually different. If you take both of those characteristics into account, they're all different. Well, no, I don't even know what is this question. You do get some random ones though. We're going to do an actual exam question next so you can see. I mean, these were based on exam questions, but obviously I've tried to... Yeah, it says man though. I was thinking it was about sheep, but then it says man. Surely it's got to be sheep. Surely this is an error. Because the spotted skin would make sense if it was sheep as well. Okay, should we do an exam question? So this is an actual exam question, okay? And I love it because it's 4 marks. And if you get it, you've got 4 marks in the bag without really having to remember any content, which is so rare for biology A level. Okay. I'm going to kind of take a back seat here and I'm going to let you have a go. Because one of the skills is reading the question and figuring out, you know, what's dominant, what's recessive, what are the parental genotypes, making sure you're using the right letters. So they have told you what letters to use. So see if you can put in the chat for me the genotype of parent 1. They've given us the phenotype. And then the genotype of parent 2. I quite often just pick key things out of the question as well. In fruit flies, allele for grey body. Oh, ebony, not black. Allele for normal wings is dominant to the allele for vestigial. Vestigial means like wings that haven't formed fully, like they're really small. So vestigial winged flies, heterozygous for grey body colour. Good, Daisy. Guys, have a go. Tells you in the question, vestigial winged flies, heterozygous for grey body colour, were crossed with ebony bodied flies, heterozygous for normal wings. How do we feel about that? I think this is the hardest one we've done because we are working out the genotypes ourselves, right? Good. Good, Felicity. Good, Tabor. Okay. Right, gametes. Do you think you could do this without me underlining? And I would put them in circles. So Grace, yeah. Oh, where are you getting the capital N from? Is that parent 1 gametes, Grace, or parent 2? Yeah. Good. Now, I'm only going to bother putting 2. I know we could do 4, but you would just end up repeating yourselves. Do you see what I mean? Like, because if you say this goes with this or this goes with this, that would give you the same gamete twice. And then this goes with this or this goes with this, that would give you the same gamete twice. Because this individual is homozygous for the wing gene. Same for this parent. I could say this goes with this or this goes with this. But then I'd go, this goes with this, which would give me the same gamete, and this goes with this, which would give me the same gamete. So I'm only going to bother writing 2. And then I can't fit it on down there, so I'm going to do it up here. And it's quite nice because my Punnett square doesn't have to be massive, because each parent can actually only make 2 types of gamete. So offspring genotypes, quite often in the exam, it does this, right? It gives you a line to write them on. So please do that. Don't just leave it in the table. Actually write them out and check you're writing them out correctly. Okay. And then the reason they put it on a line like that is so that when you write the phenotypes, they can check that you are matching the phenotype correctly to the correct genotype. So whatever order you write the genotypes in on this line, make sure you put the right phenotype in the same order underneath. So they're going to check that this genotype matches to the first phenotype that you write, and this genotype matches to the second. Yeah. So what is the phenotype of this first offspring, please? What is the phenotype of this first offspring? Because I'm going to see if we can do a ratio as well. Yeah, grey normal. I'm just going to write grey normal. You know what I mean though, right? Grey body, normal wings. What about the second one? Yeah, good. Yeah, it's ebony normal. And then we've got grey vestigial, which is basically underdeveloped wings. And we've got ebony vestigial. So what's the ratio? It hasn't actually asked me here, but quite often for the final marking point, it would say, give the phenotypic ratio. So the ratio of phenotypes. Good, well done, guys. Yeah. They're all different, aren't they? Okay. Now that was a proper A-level exam question, and that was worth 4 marks. You didn't have to give the ratio. You would literally get a mark for getting the parental genotypes correct, a mark for getting the gametes correct, a mark for getting the offspring genotypes correct, and a mark for getting the phenotypes correct. Okay. Right, that was our last question. So I've actually got a few extra slides today. I was going to give you some information about the full course. I think a few of you have asked me already. So if you want to stay on, I can just answer any questions that you might have if you're thinking of joining us in September. I've got information on pricing and things like that. So I'll just kind of talk through these slides and I'll explain as best I can what the full course involves. And then at the end, if you want to ask me any questions, if I've not answered anything, just go ahead and ask. So basically the year 13 course starts in September. I think it starts the 9th of September. It runs all the way through till the end of May. And for year 13, there are 2 lessons every week, and each lesson is an hour and a half. Okay, so 3 hours of biology with me each week. So in total, you would get 108 hours of lessons. Each lesson has a homework, which is longer than the summer school homeworks. So it's like a set of exam questions basically. And each homework, I film a video that you can then watch, and I talk you through how to do each question, how to pick up all the marks. So the videos are really good, okay? Now, the idea is that you attend every lesson, you fill in the workbook, you do the homework, you watch the video solutions. But if you miss a lesson, all the lessons are recorded. So, and they're all saved like for the whole year. So you can go back and watch any lessons that you've missed. Even if you were there for the live lesson, but you just want to go back and watch it again for revision, They're all saved, okay? You also get at the end of each month, you get a monthly assessment, which is more exam questions, different exam questions on all of the content that we've covered that month, okay? So that's how the course works. There are two packages. There's like the standard package and the pro package. With the standard package, you pretty much get everything. You get the lessons, the workbooks, the homeworks, the video solutions, the monthly assessments. The only thing you don't get with the standard package is the personal mentor. Do you guys know what the personal mentors are? Personal mentors are like university students that are doing like medicine or some kind of biology degree, and they all got A's and A stars in their A levels, and they basically are just at your beck and call. You message them whenever you want, you ask them for help, you ask them for revision tips, they can send you packs of questions, they can help you after a lesson. You just basically chat with them whenever you want. But that's on the pro package. So you can opt, if you think that extra support is going to be valuable, you can get pro package, or standard package doesn't have the mentor, but it does have everything else. Will lessons be on during the half term? Yeah, they pretty much run non-stop, apart from, I think there's 2 weeks over Christmas where they don't run. But other than that, they run over October half term, they run over Feb half term, they run in Easter. But they're all after school, so they're in the evenings. I've actually got a timetable that I can show you as well. I'm not going to talk too much about this, but this is just, I don't know if any of you have had a private tutor before. It's basically ridiculously expensive. I used to be a private tutor and I used to charge £50 an hour. And that's up north. Some of you probably live in London, so it's probably more like £80 or £100 an hour. And you can end up spending thousands. This is how much it would cost you if you had a personal tutor for 2 lessons a week for a year. It would end up costing you or your parents like £4,800, which is ridiculous. So we don't want you to have to pay that. And the other thing that some of the things that we've had fed back to us with regards to private tutors is you can't always get the time that you want. You're often not sure how good they are, how qualified they are, whether or not they've worked with exam boards. You sometimes don't get many resources from them. Like with us, you get a workbook for each lesson, you get a homework for each lesson, and you get a monthly assessment at the end of every month. So yeah, so you get a lot, which quite often you don't get with private tutors. So this is basically why we created the company, My Headspace, because we want to be able to give you the best teaching experience for as low a price as we can. Okay, I'll come back to your question, Zaina, in a second. So in terms of the value, if you went with a private tutor, on average, you're looking at spending like £4,800 a year. Like there's no way most people can afford that. So with My Headspace, these are our normal prices, okay? It's usually about £800 to £1,000 per year for the full course. And remember, that is 3 hours a week, every week, basically. But because you've been in summer school, we are offering you guys 40% off. So let me just show you what that means. You need to look at the orange box, okay? This is pretty amazing. Like, I don't mind trying to sell this to you because it is a really, really good deal. Like, I'm not a salesperson at all. But if I was doing my A-levels, I for sure would have done this. So you can pay £397 and get the whole year's worth of biology tuition. Or it's £597. I think that's the pro package if you want a personal mentor as well. Yeah, this is a special discount just because you've been coming to all of our summer school lessons. So £397. I think I've kind of been through everything that includes, but you get 3 hours a week of lessons, 2 lessons, 1.5 hours each. You get the workbook for each lesson, you get a homework for each lesson, you get video solutions for each homework, and you get a monthly assessment. And if you want the pro package, you can also get a personal mentor. That is all you pay, and it takes you from September through to May. I'll talk more about that at the end, how to actually get the deal if anyone is interested. But the other thing, just to maybe if your parents are a little bit unsure, if you start in September, there is a 14-day like money back guarantee. So if you start and after the first couple of lessons you think, oh, this isn't for me, or for whatever reason maybe you can't attend anymore, you can get all your money back. Okay, so there's no risk to you guys. The other thing that I just wanted to mention as well, we also, if you join up, you also get like an Easter revision course, which would be free to you guys, and you get cram courses the day before each of your exams next summer. And because if you join our full course, you get all that for free. So as well as the normal lessons, you'll get like 3 intense lessons over Easter for biology, and you'll get an intense lesson the night before paper 1, the night before paper 2, and the night before paper 3, and that's all included on top of the normal lessons. This is just if you want to know about any other subjects. Would this work with any exam board? What exam board do you do? Because for year 13, we've got AQA and OCR. What do you mean CIE? I only really know of AQA, OCR, and Edexcel. So for year 13, we've got an AQA course and an OCRA course. But yeah, come back to me on that, let me know. This is just if you wanted other subjects. We've obviously got maths, you probably know Neil. We've got physics with Brooke. We've got chemistry with LaJoy. And we've got a uni, like a UCAS prep course with Louis, who's the head of English. Yeah, we do them separately for AQA and OCR. Yeah, they're on at different times, slightly different times. So I don't know whether anyone's interested in this, but if you wanted the ultimate pass, it depends what subjects you're doing, right? But say you're doing biology, chemistry, maths, or physics, biology, maths, whatever. If you want access to 3 subjects, you can go for the ultimate pass, which is 3 subjects all year for like £977. So you'd have like 3 hours of math lessons, 3 hours of biology lessons, and 3 hours of chemistry lessons, or whatever subject you do. Unfortunately, we don't have like history or geography or RS or anything. But if you do science or maths and you want 3 subjects, you can get all 3 for this price, which basically takes the price down to about £2 per hour of lesson time, or £4-ish if you get the pro package. You can pay in installments, by the way, so you don't have to fork out all the money. You can split it into like 4 installments. So this would be for the ultimate pass, this price. If you wanted like 3 subjects, you could just pay £244 today, and then you'd have 3 more installments. But that's for the ultimate pass. So that's if you want like 3 subjects for A-level. What if you do 2 subjects? The best thing to do would be to contact the team and see if they can offer you like a deal. If you want to do more than 1 but not 3, they might be able to do you some kind of deal. I would just, to be honest, I would just contact them anyway. Even if I wasn't sure, I'd just get in touch and just ask my questions and just see, because they're really good. The customer service team are really good at like figuring out if the course is suitable for you, if the exam board is suitable for you, like what the best offer is they can do, if they can do any discounts. You just get in touch, guys. What you'd have to do, Aaliyah, we don't have a biology specific A-level course for that exam board. Okay, so what I would recommend you do is print off your have you got your specification, Aaliyah? Have you got your spec printed out already, or could you find your spec online? Because what you could do is compare your spec. It will be very similar. Like AQA and OCR are very similar. So I would compare it and see how similar it is. Because I've got to be honest, we don't have a course specifically for that exam board, but it's very likely that the majority of the content on that course is the same as AQA or OCR. So you could find which one it was most similar to. That would be my advice if you want to join, yeah? So in the description for this lesson on YouTube, you can tell I'm not great with tech, but just in the description, there should be like a little link that says like summer offer or something like that, or summer discount. If you click that, it will automatically take you to WhatsApp. I think it even fills in the word summer 2024. Someone can try it now and let me know. You send that message, it sends it through to our customer service and sales team. And that will basically tell them that you have been in summer school, so you can get 40% off. And they will then start a chat with you. That's all it is. So I would just do it. Even if you're still not 100% sure, or you've got questions about subjects and combinations, I would just do it, because it will just get you into a chat, and then they will be able to advise you and give you like a confirmation of price and stuff, and yeah, and get you sorted basically. Yeah, with chemistry, there's AQA and OCR, but I've got a timetable actually. So shall I put that on the screen? The OCR biology one isn't on there, but we do have an OCR biology one. It's just, I think OCR biology is going to be Monday and Tuesday. So I can leave this on the screen. But chemistry, yeah, there's chemistry OCR and chemistry AQA. There's biology AQA, which is on here. There's also biology OCR, which is going to be Monday and Tuesday. You can see when the chemistry, maths and physics is. Zaina, what was your question? Just remind me what your question was. Do you go over all the content, or only year 13 or year 12? So that's a really good question. You both asked similar questions there. So the year 13 course, the way it works is we spend most of the year going over year 13 content, but the last couple of months, we revise year 12 content, and we also revise year 12 content in Easter, during the Easter revision lessons, and obviously the night before paper 1. You could ask about getting access to the year 12 course as well. If you wanted to literally do everything again in year 12 from scratch, you could ask if you can get the year 13 and the year 12 course, and you could do both of them. Yeah, or you could do the year 13 course and just do the year 12 revision with me at the end of the year 13 course. But yeah, it's a thing you could ask. I would send a message in and say, look, I want year 13, but I also want access to year 12 lessons, and see what they say. There's no harm in asking. I was going to ask if I should do the course even though I do Edexcel. So again, I'd have to say the same to you, Zaina, as I said to Aaliyah. It's not Edexcel specific. However, I'm a little bit more familiar with Edexcel than what Aaliyah is doing. So I know that there's a lot of overlap between Edexcel and AQA. But I would recommend, if you're thinking this would be useful, I would recommend getting your spec and comparing it to AQA. I'd say about 80 to 85% of it is the same, but it might be that there's just like a few topics that we do that you don't have to do. So you might like miss those lessons, I suppose. Yeah, and it might be in a slightly different order, but I don't want to like promise you it's going to be exactly the same. What I would do is get your spec and compare it to AQA, just in case there's like one particular topic on your spec that we don't end up covering. And at least then you'll know that you're going to have to revise that bit yourself. Yeah, minor subtopics and wordings. I think it would still be useful, to be honest, Zaina, but that would be your decision. What exam board is maths? As far as I know, Saya, the maths is pretty generic. I think it's mostly aimed at Edexcel, but I know that Neil has said before that the maths exam boards are so similar that his course is pretty much applicable for all of them. But again, you could message the team and ask. Is there any other questions, guys? Is there anything that I've not answered that you're still curious about? It is like I said before, I'm not a salesperson. You know, it's not particularly comfortable for me trying to sell this to you, but it is the best value for money. Like, you're not going to get better value for money. Like, if you just do biology, what did it say just for biology? I can't remember how much it was. £397 for one course. So that's for the year. That's for like 9 months of lessons, 3 hours a week, and all your homeworks and monthly assessments. So it is pretty crazy. Remember, this price is if you do 3 subjects. How many of you do like 3 subjects, maths and science subjects? Because that's the price. 977 is without the mentor, I think. And I think 1477 is with the personal mentor. But that would be for 3 subjects. Yeah, so Sam, it might be useful. Like, if you want to do chemistry, biology and maths, you could get that for 977, do all 3. You'd have a pretty hefty timetable. Yeah, chem, maths and bio. So you might want to opt for that. It's up to you. It might be that you're actually only struggling in one subject. You just want one subject. The installments, I don't know exactly how it works, but they take this price, so 977, they split it into 4. There you go. So you'd pay, like, your first payment would be 244, and then I don't know when you'd pay the next one, but they'd split the payments up into 4. Will these lessons go through all the content of year 13? Yes. I mean, I can even show you. This is how, let me show you guys. I probably shouldn't show you this, but I'm going to. I don't really mind. I'm going to show you this. Put it on my screen. Can you see this? I don't know, it's probably too small. Let me make it bigger. You can see, like, we've already done a lot of planning. So yeah, you go through the whole course, even the required practicals. Okay. So you can see I've broken it down. Each lesson. Where's what we've done today? Dihybrid crosses. There you go. In January. Yeah, the whole of year 13. We even go through required practicals, practical questions, maths questions. And then you can see, like, I'm going to do a lesson purely on statistics. I'm going to do a lesson purely on essay writing. I'm going to do a load of year 12 revision lessons, like in April and May. These are the 3 cram courses, so one before the night of each paper. And don't forget, on top of this is the Easter revision course as well. That's not even on here. And Easter revision, we do like about 6 hours of year 12 revision. Yeah, there are specific OCR lessons as well. This is the AQA one, but there is the same thing for OCR. But obviously OCR, the topic orders are a little bit different. The content's a little bit different. This is the AQA one. But there is an OCR one. Yeah, definitely go over question techniques for sure. Yeah. 9 markers. We don't do 9 markers. On AQA, the most we do is a 6 marker, and then we do a 25 mark essay. So I'm guessing if you're asking for 9 markers, you're doing a different exam board. The mentor, yeah, so the mentors, so Neve was actually in this lesson. I'm not sure if she's still here. But the mentors are university students. They're quite often studying medicine or other biology related degrees. They got A stars in their biology A level. And basically you have open communication with them. So we use Discord. I don't know if you've heard of Discord. I'm sure you have. And they basically just check in with you. So they check in with you, they ask you how it's going, they ask you how the lesson went, if there's anything they can help you with. You can ask them questions about the homework, you can ask them questions about the lesson. But what I think the best thing was last year is the mentors would quite often say, look, here's a pack of questions with dihybrid crosses. Here's a pack of questions on structure of the heart. And they would send lots of extra questions. And students would even like send them screenshots of their answers so that the mentors could mark them for them. You could send the mentors like essays. You could send them screenshots of essays that you've written and they can try and mark those for you. It's basically whatever you need. If you want uni advice, career advice, you can pretty much message them anytime. They get back to the students really, really quickly. They're just there to support you and help you as much as possible. Yeah, that's the mentors. Is the installment only for the ultimate pass? I'm not actually sure, Jay. What it would be best to do if you don't want the ultimate pass and you just want one course, like the orange box, I would just I'd send the code word anyway and just say like, I just want one course, I just want to do biology or whatever. Can I pay in installments? Okay. Yeah, Natasha, don't worry. OCRA, we've definitely got a course for OCRA. It's specific because last year we tried to do OCR and AQA together and it didn't quite work because there are some topics that kind of didn't quite line up. So there's a totally separate course. So in the chat, if you go now, oh, also, can you like this lesson? Because this is on YouTube for you guys. So can you like it? I would really appreciate that. If you're still here, I should have said it before. Yeah, make sure you like the lesson, but then just underneath, there's a little link in the description. I think it says something like discount or summer offer. If you click it, it should take you straight through to WhatsApp. I have a personal tutor with teachers through emails and I should really use them more, but so our mentors are over messages as well. So if it's not the kind of thing you've been using, you could always opt to not have that pro package. Okay. Right. Anyway, I'm not going to keep you any longer because you've stayed 20 minutes past the lesson. But hopefully that's been useful. It's given you some food for thought. And I'm hoping that some of you do join me because the students that are most successful with us are the ones that start right in the beginning, that start right in September. We have a lot of students that kind of just come for the cram course or just come for Easter revision. And they always say, I wish we'd joined earlier. Like I wish we'd been doing it all year. So this is your opportunity to kind of make that positive step and get it for the cheapest price because this price won't be available if you wait until the course has already started. Okay, so this is just for summer school students. Okay, thank you so much. I'm going to see you again next Thursday, so have a great weekend and I will see you next week. Don't forget to do your homework. Thank you. Bye, guys.